Thank you very much. Thank you very much for the introduction, and thank you, organizers, for the kind invitation. Okay, so what I would like to do in this short time, like we were just introduced, get a bit of a general overview of our field, cellular senescence, its role in aging, and what our lab is doing in this field, and a few words about our recent studies and where we are going and what we are interested in. So if we talk about aging, all know that there is, after a certain age, exponential increase in the probability to get different age-related diseases. And this is part of the aging phenotype. But how we start to understand it, if we want to understand it, we need to understand the underlying molecular mechanism and also underlying cellular mechanism out of these processes. And this, over the years, were called hallmarks of aging, and there are quite a few such a hallmark. We, in the lab, are particularly interested in one of them, called cellular senescence, here. And of course, all these hallmarks are interconnected by the variety of molecular pathways. So what is the kind of, in general terms, connection between senescent cells and aging? Senescent cells are normally formed in the organism, and then they're dealt by the immune cells that clear them from the tissues. And this kind of turnover maintains the normal homeostasis of tissues. With age, senescent cells tend to accumulate within tissues, and this accumulation leads to chronic inflammation, tissue destruction, and accelerates development of different age-related diseases, including neurodegenerative diseases, cancer, lung diseases, and many others. So how is this that the cells that form in our organism become detrimental? From the evolutionary point of view, nothing should be selected if it's detrimental to the organism. The fact is that senescent cells, while they're forming, they're actually beneficial. So there are a variety of different triggers that can trigger senescence process, and this is through molecular pathways, including p53 and Rb pathway. And this formation of senescent cells have beneficial short-term effects. And this is a tumor suppression because senescence is a terminal cell cycle rest of cells. So if the cell with oncogene mutation cannot proliferate anymore, it cannot form a tumor. The cells, when they are forming, limit in tissue damage because their excessive proliferation is limited and tissues can return to the pre-damaged state. And senescence process also plays a role in embryonic development in different transient structures, and this role is to guide in different cells to get to their places or to facilitate interaction between the mother and the fetus. So senescent cells are necessary when they're formed, but the bad thing happens when they accumulate in tissues. So when they accumulate and secrete what's called senescence-associated secretory phenotype, or SASP, they can promote tumorigenesis and promote tissue aging. And then immune system comes here and tries to uncouple positive short-term responses from the negative long-term responses, basically keeping the normal homeostasis by maintaining the normal turnover of senescent cells and by eliminating senescent cells after they are formed and after they have executed their beneficial function. So we can think about different ways how we can directly eliminate senescent cells, in a way mimicking the immune system or helping the immune system to eliminate senescent cells. And over the years, this kind of approaches called senolytic therapies, which are, some of them are under development. Quite almost a decade ago, we have identified one of such pathway where we could use BCL2 family inhibitors to eliminate senescent cells, and we can, and others, many other labs use this pathway to, targeting this pathway to eliminate senescent cells in the variety of models to study the role of senescence in aging in different age-related diseases. So with this kind of tools and understanding in hand, we can ask what is the role of senescent cells in tumorigenesis and in tissue aging. So specifically for tumorigenesis part, we have identified that senescent cells, and this is in the model of pancreatic cancer, that elimination of senescent cells from premalignant lesions actually limits development of pancreatic cancer. But going back to aging, a few years ago, in the model of accelerated aging due to lack of immune surveillance of senescent cells, we are able to eliminate senescent cells pharmacologically, and this extended the median lifespan of mice, suggesting kind of one of the first studies suggesting that pharmacological elimination of senescent cells can actually extend lifespan of mice. Now, this is for the general lifespan. What about specific age-related diseases? So over last several years, we have done several studies on age-related diseases, and one of them on one disease that is, I think, very familiar age-related disease, and this is Alzheimer's disease, where we identified population of senescent microglia cells. Say a student in the lab, former student Noah Rahmian, led this project, where we have identified this senescent microglia population in different models of Alzheimer's disease, and also in just simply old mice, and also using bioinformatic approaches in the single-cell RNA-seq data of human Alzheimer's brain. So we also looked at the molecular mechanism, why this senescent microglia form, and this because of TREM2, and we have looked at TREM2 knockout mice and have seen that there is a decrease in senescent microglia if the TREM2 knockout mice. One interesting thing that if we treat with the senolytic that eliminates senescent cells from the periphery, we could not only reduce amount of senescent microglia in the brain, but also observed the improved, I would say, some way cognitive function or memory function, this mice in this novel object recognition test that is depicted in this figure, where mice are asked to remember objects if they have seen it or not. And Alzheimer mice don't do this, and Alzheimer mice treated with senolytic are able to remember this object. So then we followed up with metabolomic studies on senescent cells, and I'm sure we will hear more about metabolism and aging from Chaim and others. So we did metabolomic profiling of senescent cells, and we have found significant changes in the lipid metabolism, and then we linked it to the microglia, and we have identified that senescent microglia actually contain lipid droplets, and it is known in the literature that microglia with the lipid droplets are very detrimental, and they are correlated with the worst outcome in the Alzheimer's disease. And one interesting kind of note in this study is that many, many years ago, when Alzheimer described the Alzheimer's disease, he noticed that there are many glial cells that are showing adipose sacules, which are basically lipid droplets. So these lipid droplets in microglia were observed by Alzheimer many, many years ago, and now we identify them as senescent microglia cells, and we know that they did contribute to Alzheimer's disease, and this study is now under revision. So going from the brain to the body, looking now at different age-related lung diseases, and lung diseases are very prevalent between age-related diseases, both pulmonary fibrosis and COPD. So over the years, we looked at these models, and in one of the studies, also published last year, we identified that senescent cells upregulate PD-L1, and PD-L1, as many of you know, is an important checkpoint, which is very important in cancer therapy, and there are a variety of drugs that are targeting these pathways, both PD-L1 and its ligand PD-1. So here on this graph, you actually can see that P16, one of the markers of senescence, and PD-L1 expression are very much correlated. But one interesting note is that if we're looking at, these are epithelium cells from the lung. If we look at the basal expression of PD-L1 in the old mice and in the young mice, the basal expression is very, very different, and we can see that actually PD-L1 is highly expressed in all epithelium before kind of any treatment or any correlation with PD-L1 with P16, and this has many implications, for example, for anti-PD-1, PD-L1 therapy. We also were able to use different antibodies to eliminate this P16 positive, PD-L1 positive cells from the lung. And one of the ideas is that because PD-L1 is an immunoinhibitory molecule that can inhibit elimination of senescent cells, therefore can explain why the turnover of senescent cells is inhibited in aging. So as I mentioned in the beginning, there is a normal turnover of senescent cells in the organism, so we can suggest that increase in PD-L1 and some other immunoinhibitory molecule is the mechanism that inhibits turnover of senescent cells with aging and leads to their accumulation and leads to these deleterious consequences of their accumulation that we observe. So one other pathway that we have discovered several years ago that actually important to maintain viability of senescent cells, but only this is a P21 pathway with CDK4. P21 is a CDK inhibitor, which inhibits cyclin-dependent kinase 4/6 function. So we have observed that knockdown of P21 is actually senolytic, means it reduces viability of senescent cells, but not only it reduces viability of senescent cells, it reduces profibrotic signatures in senescent cells and in many other cell types. That leads us to study role of P21 in lung fibrosis, and a student in the lab, Norit, who is here in the audience, did this beautiful study that we published, and where we used P21 genetic model for P21 knockdown in mice, and we have shown that this knockdown, which is done during the development of the disease, not before, but during, like a real treatment, actually reduces amount of senescent cells in fibrotic lung, reduces accumulation of immune cells and inflammation, and reduces fibrosis and accumulation of fibrotic tissues. So, and preserves actually the lung architecture. So these mice without or with reduced P21, which P21 was reduced during the development of the disease, actually protect the lungs from the disease development. And now, because we also understand the underlying molecular mechanism, we can try to target this mechanism and develop effective and broadly active antifibrotic agent based on this mechanism. And that's one of the things Norit is working right now. So this is obviously not the only senolytic or senotherapy that are around. There are many senolytic therapies that are proposed, and xenomorphic therapies, meaning that not simply kill senescent cells, but just reduce their impact on the other cells. And this is a very short summary of this, published last year, and also kind of mentioning on the top this inhibition of BCL2 family and many different pathways. And there are many clinical trials going on, and 30 is probably, it's from the last year and probably underestimation, and I would say now probably there are maybe 50 or 60 clinical trials going on evaluating different xenotherapeutic agents in variety of different age-related diseases. But one kind of challenge is to know, and in all these clinical trials, was it effective, not only in terms of the disease progression, but have we actually, we give senolytic drug to people, but we don't know if they actually eliminated senescent cells or not. So, and this is important question in practicality and also in the mouse experiment. So we developed a method to quantify senescent cells on the single cell level, which is good for mice and good for human samples. So we were able to make this method working, and now using this method, we can quantify senescent cells in mice, but also in PBMCs from patients. This help to calculate risk of age-related diseases because we can understand what is the relative burden of senescent cells in different individuals, and this method can also help to directly evaluate the efficacy of senolytic interventions. And we are in the process and a discussion of kind of translating it into the real life from the lab. So with this kind of in the final slide, say, okay, if we are looking at aging, and with aging there is increase in probability of many age-related diseases, we can kind of looking at this scheme divider, all the lifespan to health span, and maybe some people call sick span. So when we look at different therapies that actually want to give in terms of aging, so there are several scenarios, but I just want to show two kind of extremes. And one is just general extension of lifespan, but this general extension of lifespan can actually come up with extension of sick span. What we really would like to achieve, I think, and where we really need to aim is to the extension of health span and not only lifespan, or not necessarily maximal lifespan, but really to the extension of health span, and this I believe would be most beneficial for the people. With this, I would like to thank, first of all, my lab members who did all the work that I mentioned, my lab members over the years, and people who did the work that I mentioned today. And here we are in Jerusalem, and to Jerusalem, also our collaborators who help us with many studies, and many other collaborators, and not less important, the funding sources and supporters who made it possible to do this big experiments. So thank you very much.
Thank you, Valery, for this excellent talk. The question is regarding the Alzheimer's mice where you treated with senolytic drugs, right, and they got better. So my question is, did you see any reduction in senescent microglia or senescent astrocytes in the brain, or what actually could be the reason for the reduced pathologies that you observed? Okay, so yes. Basically you give the drug systemically, all the mass was affected, so what was actually given this, reduced pathology?
Yeah, so we indeed have shown reduction in presence of senescent microglia in the brain. This being said, we don't know if the effect on the senescent microglia by the drug is direct, because theoretically drug is not supposed to cross BBB. So what we think is happening is that the drug, like it was shown many times before, is active in the periphery and reduces the amount of senescent cells in other tissues, including the circulation, and this is what affects the amount of senescent microglia in the brain. We cannot exclude that because BBB is somewhat damaged in these mice, right, because of the disease, then some of the drug could get into the brain. We actually did all kind of experiments trying to assess by mass spec if we have drug in the brain, where in the brain. But these are, we were not able to detect high levels of the drug in the brain by mass spec.
The test you mentioned to measure senescent cells, is that commercially available, or is that a tool that you use just inside your institute?
It's the tool that we used inside the institute, but we are working now with a commercial entity to translate it to the real life, as I mentioned, and this process is ongoing. So if you are interested, we can discuss it, right? So I've seen there are many more questions, and I will be more than happy to answer them during the break. Thank you very much.
Yes, thank you so much.